today I don't feel like taking it.
Remember, my videos are always welcome. Oh wait, I need a pencil. When the sun and she comes, can I borrow your pencil? Because my pencil's somewhere deep in the cavern of my bag.
Do you want a pencil now so then you can...
That'd be really nice.
You mind a pen?
Yeah, that's fine. Just writing 2 initials. That's at least something I can't mess up.
Yeah.
Have
You get the exam and write the wrong name.
Have you ever accidentally misspelled your own name?
Yes.
Alright.
I think I do it all the time. And do it when I'm like thinking of something, or I'm writing my name and I'm looking at the next question. And then I look up and I'm like,
Yeah. That's why pencils are good friends.
I do that a lot. That's what it is. I do that. Way too much.
I misspell my last name of Brown sometimes. I'm like, what did I even try? What did I try and say? Well, and I mix up the letters in my name. Letters. They are complicated business there.
End of my existence.
I'm not sure how I passed kindergarten.
How am I here?
They apparently put me in a gifted class after a small test in kindergarten.
Really?
Yeah. It was like once a week.
Tested really bad. I would always be put in like the really bad group, and then I would just like by the end of the semester I'd work my way up to the good group. As teachers would like slowly realize like, she doesn't need to be here.
I was just. I think they did it because I was creative. Not the intelligential thought, but I was creative.
Yeah. That didn't work for me.
Although I eventually became smart, I was 4th in my class.
Dang. Really?
Yeah. And the valedictorian and the salutatorian were both in the gifted class with me. 3rd place just came out of nowhere. She didn't even come to half our like physics classes. It's like I'm very surprised. Well, enough about me and my smartitude for a little bit.
Sorry, I'm just people are like texting me about meeting and whatever.
That's all good. Oh yeah. My advisor called and said I actually have to go and meet them before I schedule my classes now.
Oh really? Why?
I don't know, probably because I was tough.
Get started. We don't have too many slides and information left, and we will do a review. Last time we had looked at DNA replication and how that takes place, talked about the enzymes and so forth, talked about that replication complex. And this shows basically what's happening where we're having the 2 strands copy, complementary copies. It shows some of the enzymes that are involved. As I said the last time, I really don't get in that much on what DNA polymerase 1, 2, and 3, they all are involved. They come in at different points. Just remember that the enzyme responsible for the elongation of that strand will be the DNA polymerase. They make the DNA strand. Helicase is the enzyme that opens it up. And remember, we must have a primer that is made of RNA to get started. It's just about 10 to 15 bases, nucleotide bases long, not very large. Note also that there are transcription factors on there, proteins that help to stabilize the DNA while this is occurring. But there are other proteins that play a role. So that was the basic setup right there. And now we're going to look at a part, how does when DNA is copied, there is always the possibility that a mistake can be made. Those mistakes would be a mutation. Mutations can be because a nucleotide is substituted. It can be because a couple are switched. It can be because a nucleotide is left out. Now, one of the things you guys will find when you take genetics is, we get into this a little bit deeper. If you note the codons, remember there is a 3 nucleotide sequence that determines what amino acid that is going to be used. That is called a codon. There can be nucleotide substitutions made, and don't try to remember this. This is just to help you understand. I'm not asking you this on the test. But those types of substitutions, and your book's not talking about this, are called non-synonymous or synonymous substitution. And basically what that means, if the substitution in that 3 codon set of nucleotides, if the substitution occurs on the first or second nucleotide, it will change the amino acid. Now, if you change the amino acid, you've just made your protein non-functional. That usually results in a fatal circumstance for the organism. A synonymous substitution, if it is made on the 3rd codon, it really does not affect the amino acid sequence. So you can have people who have the same protein, but have a little bit different code, particularly on that 3rd, a nucleotide substitution. That does occur. It's passed down through generation to generation, particularly on mitochondrial DNA or the Y chromosome. And those substitutions are what's used in history to determine if you've got this one group of people that's lived in this part of the world, they've not intermixed with other groups, they will replicate these substitutions over and over. And we can say, because you've got that substitution, these particular ones, then you're related to this group of people. That's the way ancestry works. But the key, it doesn't change the phenotype. It's on a nucleotide, that 3rd one, for whatever reason, I don't know the reason, but for whatever reason on that 3rd, because of the other codes that can be there, it does not change the amino acid sequence. The first 2 does. So that's how we can have mutations occur and not kill the organism, or not be fatal, or cause a disease. The phenotype actually stays the same, but the code is a little different there. There is flexibility within that code. Now, whenever there is something that goes wrong, the incorrect nucleotide is either placed, or if a nucleotide is left out, DNA tries to repair that. It has a mechanism. And notice up here, it says the DNA polymerases proofread newly made DNA. So these are the enzymes that do the proofreading. They can replace any incorrect nucleotides, and they are pretty good. They have a good success rate. In mismatched DNA, repair enzymes correct the errors in those base pairs. Note also, DNA can be damaged by exposure to harmful chemical or physical agents such as cigarette smoke and X-rays. It can also undergo spontaneous changes for no particular reason. There was an article in some of the news papers came out today, and it is showing that wolves that are in the area of Chernobyl, where the nuclear leakage occurred in Russia, those wolves are showing those mutations now. They're having differences in things in the way they behave and in their phenotype. So, X-rays, radiation can cause that. Anything that can damage that DNA, chemicals, whatever. Sometimes it just happens abnormally. many times, depending on where these mutations occur, if DNA does not check them, you will get perhaps cells formed that are not normal. They can be the beginning of a cancer cell. It is then up to your immune system to locate those abnormal cells and remove it. If they do, you never know they were ever there. If they don't, then you can get cancer. And that's one of the things cancer cells do. Remember, is they have uncontrolled replication. Because of their makeup, they don't submit to the body's normal restrictions on cells and proteins they can hide. So, it says here, in nucleotide excision repair, a nuclease cuts out, replaces damaged stretches of DNA. This shows that happening. DNA polymerase will actually read, and then those nucleases, if there is an incorrect segment, can replace that. DNA ligase will then tie it back together. That will enable the bonds between, hydrogen bonds between those spaces and the phosphates and the sugar to occur and tie it back together. Ligase, when you see that, think of the term ligature, to tie. That's where that comes from, ligase, to ligate. Limitations of this polymerase exist. The usual replication machinery provides no way to complete the 5' ends. So there are some sections that this would be difficult for it to do. So repeated rounds of replication produce shorter DNA molecules with uneven ends. This may be part of the things when we talked about chromosomes like the telomeres. Those ends, they shorten as age occurs whenever your system and your machinery is not functioning exactly right. Those are the telomeres, those ends of DNA. They are thought to act just like the little plastic tips do on the inner shoelaces. They're there to hold everything together and to protect it, the main part of the shoelace. Those telomeres are the little caps on the end of DNA that are there to protect it. Notice it says they postpone the erosion of the genes near the ends of the DNA molecules. They are a protective device. It has been proposed that the shortening of telomeres is connected in some way to aging. Your machinery, biological machinery, just not, or losing its ability to completely, accurately replicate. An enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. So what that means is, it's a way to try to counteract the shortening of the telomeres, particularly as the cells become shorter in every cycle. Essentially, genes would be missing. So to prevent that from happening, telomerase tries to maintain those ends and make sure that they stay functional. But as age comes on, their effectiveness is reduced. So as people get older, there is a shortening of chromosomes. Notice, this shortening of telomeres might protect cells from cancerous growth by limiting the number of cell divisions. There is evidence that telomerase activity in cancer cells, which may allow cancer cells to persist. So it's kind of, it does some good, but it does some bad too. So, a chromosome consists of a DNA molecule packed together with proteins. Now, we've already talked about chromosomes. Hopefully, you already know most of this. says eukaryotic chromosomes have linear DNA molecules passed or associated with a large amount of protein. That's the chromatin and all of the different types of chromatin that we've talked about. And they remember chromosomes appear when the cell's getting ready for replication. Most times, there's no chromosomes in the cell. It's the DNA that is in a linear form. One of the differences between us and bacteria, in bacteria, they basically have one big chromosome that's a circle. They're circular. Notice it says the DNA is supercoiled and found in the region of the cell called the nucleoid. Chromatin, that complex of DNA and protein, is found in the nucleus of the eukaryotic cells. They fit into the nucleus in an elaborate multi-level system of packing. In other words, they're condensed. This shows this process of taking DNA and turning it into a chromosome. It acts with these little particles here, histones. Notice how they will come together and start to bind. They will coil that. And eventually it's almost like knitting. They knit a coat for the chromosome, those histones. And also, again, remember that enzymes, proteins are what's doing this. And notice down here it shows you this is a metaphase chromosome. The reason that this occurs only in cell division is that DNA has to be exposed in order for us to make that DNA functional, to make copies, messenger RNA, to make proteins. So whenever we're getting ready to divide a cell, we want to protect that sequence, the genes, the nucleotides, the structure, and that is protected by stabilizing it with this heterochromatin or histones. Chromosome undergoes changes in packing during the cell cycle and interphase. Some chromosome is organized into a 10 nanometer fiber, millions of a meter, but is compacted into a 30 nanometer fiber through folding. Through interphase, chromosomes are not highly condensed. They still occupy specific restricted regions in the nucleus. One of the reasons I don't like this book, this would have been much more valuable information when we were getting ready to start to learn chromosomes instead of covering chromosomes, and then chapter 16 you cover this, which I intend on correcting. But this is telling you what is happening during interphase in copy DNA. This shows you a karyotype. A karyotype is a picture version of all the chromosomes in an organism's body. Notice at the top, these are not pairs, are they? I mean, they are one from your mother, one from your father, but they've not been copied, right? So this is showing you chromosomes that have not yet been copied into the X form, where they're together. The 23rd chromosome will be the sex chromosomes, whether you have either an X or a Y. This is not the way chromosomes appear in cells. I mean, they're not ordered. They're like this. Now, they will have their mates, like you can see there, but they are all going to be in the nucleus, and it's not going to be evenly separated, numbered 1 through 23. Most chromatin is loosely packed in the nucleus during interphase and condenses prior to mitosis. loosely packed chromatin is called euchromatin. During interphase, a few regions of chromatin, centromeres, and telomeres are highly condensed into heterochromatin. Dense packing of the heterochromatin makes it difficult for the cell to express genetic information coded in these regions. So, DNA is not packed, it has to be open so that it can be functional. We can make proteins. It says to express genetic information, that means to make proteins. Histones can undergo chemical modifications that result in changes in chromatin organization. So,
I do have a question about the nucleoid. Okay, wait, I'm thinking of the nucleolus. Never mind, I answered my question.
nucleoid is going to be a place in a nucleus that is where the actual DNA is in the nucleus.
So that's for the bacteria, right? And then the nucleolus.
There's a nucleoid in a bacteria.
Okay, and then the nucleolus is for us. It's in our nucleus in the eukaryotes.
We've got the nucleus and the nucleolus. And other is in the bacteria.
Okay,
But they're almost, the difference is bacteria doesn't have a nucleus.
right.
Nucleoid is the kind of thing, it's nucleus-like, but it's not a nucleus, doesn't have a membrane, it's not protected or surrounded. In our cells, euchromatin or eukaryotic cell, we've got the nucleus. The nucleus is filled with a substance called nucleoplasm. In the nucleoplasm will be these enzymes and the nucleotides and things to copy to make RNA. It will be a storage area, resources. The actual chromosomes will be in the nucleolus. That is the condensed area.
Okay, I see. Thank you.
Now, go back. I want to mention some of the types of things that we've talked about. First, with mitosis, remember, mitosis is not that much different in some ways than, or meiosis is not that much different than mitosis in the fact we go through the interphase and we go through anaphase and metaphase and all of that. There are some differences though. It's not so much that there's a actual difference in the cycles as much as some sections are repeated back and forth and there's a few added, like crossover. So let's look at those. Remember, mitosis produces diploid somatic cells. Meiosis or meiosis produce gametes, the egg or the sperm. They are haploid. They only have one set of chromosomes. How many sets that an organism had is called its N number, small case n. So if you are asked, once again, what is the chromosome number of a haploid cell, it should just be N. N represents one set. If it's a diploid cell, it would be represented as 2N. When you're asked the chromosome number, remember you are being asked how many sets of chromosomes, not how many individual chromosomes. And everybody should remember also, every gene is going to be at a particular place on the chromosome that is called the locus. And most of the DNA will be packed into chromosomes. Now, they will be some stages loosely packed with a little bit of the round called the euchromatin. Sometimes it will start to condense more, make the connections between chromatids, heterochromosomes. But when we get into cell replication, they are the full-fledged chromosomes. We mentioned asexual reproduction. I'm not going to cover a lot about asexual reproduction on this because you're going to get a boatload of that whenever you get into plant study next semester. There'll be a different type of asexual reproduction cycle, alternation of generations for ferns, one type for moths. It will refer to, is this, what's the adult stage of the plant? These are the life cycles that we're talking about. And we mentioned what a karyotype is in order to display the chromosomes from the cell. Somatic cells are the ones that will be multiplied by mitosis. When you have two chromosomes in each pair, the two chromosomes that make up a pair, they're called homologous, which means the chromosome for your mother and the one from your father are the same. They just have different traits, values, but they are the same. They have the same genes and the same locus, and so forth. Every chromosome does not have the same number of genes. We've got about 14,000 genes, and they are spread throughout these 23 pairs. So chromosome 1 will have traits and genes that you won't find on chromosome 2 set. These shows, once again, these copied. Remember, these are called sister chromatids because each chromosome here is being copied. These are what makes what we call the X shape of chromosomes. Remember, the first 22 chromosomes are called autosomes because they are the same, they code for the same. The 23rd pair in people are called the sex chromosomes because they determine male or female. Remember, on male and female, they code, these are not autosomes, they do not code for the same thing. Basically, the X controls primarily the physiology of the female and for reproduction and similarities on the Y chromosome for males. Yes, as we've already talked about diploid cells and so forth, the sister chromosomes and haploid cells, you do need to remember that first fertilized cell is called a zygote. That is the first cell in an organism in which it has the complete set of chromosomes. It is, once the zygote is formed from the egg and the sperm, then it starts to multiply by meiosis, or mitosis. Meiosis makes the egg and the sperm. After fertilization, the cell division is mitosis. In plants, we have the alternation of generations. And the basic process is the same, but it will differ or vary based upon the type of plant that you've got. But if we start out here, if we have an egg and a sperm, and they fertilize, become a zygote, then they have the full chromosome number to the end. Now this is showing you in a mouse, in a diploid organism. That zygote multiplies until we get the mouse. We take some of these diploid cells and then we can go through meiosis and make the egg and the sperm. Pretty simple, straightforward. In plants, it's a little bit different because these individuals can grow up to a structure that we will see and can be the dominant structure. That is called alternation of generations. And there are two versions. Alternation means you can have an adult that is haploid or an adult that is diploid. The diploid version is called a sporophyte. That would be like us if we were the sporophyte, we will make eggs or sperm, the gametes, and those gametes will be haploid. They will unite to make the diploid organism. In plants, it can be a little bit different. The sporophyte makes haploid spores like we make sperm and an egg, but these spores grow by mitosis now. We've got a haploid spore, and they grow by mitosis into a plant structure called a gametophyte. The gametophyte makes haploid gametes now, not spores. The spores are basically just cells. Those spores grow into a structure that will then make the egg and the sperm. The sporophyte makes haploid spores. Those spores grow into a structure called the gametophyte. The gametophyte will then make the egg and the sperm. Haploid. Fertilization then of those gametes results in the diploid sporophyte. So if we go back to that structure or picture again, here we got egg and a sperm. They fertilize, they make the zygote just like in animals. Then they go through meiosis just like in animals. But the difference is when these go through meiosis, they don't make egg and sperm, they make spores. These spores are a haploid number. They can grow into a structure called the gametophyte. Then the gametophyte makes the egg. So the difference is, instead of like in animals, we just produce egg and a sperm, plants first produce a spore that will be haploid, that will grow into a structure that will then produce the egg and the sperm. Some plants, as you'll find in 1082, the adult version on most plants is the sporophyte. Some, the adult version, like in moss, will be a gametophyte. But they will get that into you. I just want you to have an understanding of what a sporophyte is, what is a gametophyte, and what do they grow into.
Do you mean by what they produce? Oh, right. So sporophytes, spores, which then become, then grows the gametophyte.
Gametophyte produce gametes.
Gametes. Right. So that's basically it. Okay, thank you.
And just understand that when the spores are produced, they're haploid. But whereas in animals, those haploids have to come together to make the organism, they don't in plants. They can grow into a reproductive structure called the gametophyte. And then some of those gametophytes can, as you'll find out, can be male that will produce the sperm. Some will be female that will produce the egg.
So they just stay haploid. They don't have to turn into diploid. They just
grow
okay.
into that structure. And then the gametophyte produces haploid cells again, except this time the cell's not a spore, it's an actual gamete that can be fertilized to grow into the diploid structure.
Okay.
Sporophytes themselves are diploid, but they produce haploid spores through meiosis. I'm not going to go into it any deeper than that because, like I said, there's differences where this occurs, and you get into that quite heavily in 1082. About meiosis itself in general. One of the differences is mitosis has one cell division. We take one cell that's going to divide and become two cells, both diploid. In meiosis, that occurs, but then there is a second phase in which those two cells produced, those two diploid cells, now separate and become haploid into four cells. So there are two cell divisions. Meiosis produces four cells, mitosis produces two. Meiosis produces cells, these four cells, that are haploid. Mitosis produces diploid. This tells you what in general happens most is 1 and 2. Homolog spare separate, resulting in 2 haploid daughter cells with replicated chromosomes. So, while mitotic cells are diploid, these are haploid. Okay, like think of it as we've got an egg, okay, not been fertilized yet, so it's a haploid cell, right? We duplicate those chromosomes. So we've got 2 cells now, haploid still a number, but they have a copy of those haploid cells. 1 copy stays in those 2, 1 moves to others, so we wind up with 4 cells. Showing it right here, we've got in meiosis, this is interphase where they're copied. In meiosis, we don't have 2 sets of chromosomes here on number 1, for example. We only have 1 because it's haploid. These 2 chromosomes that have been copied just go into different cells, and the other one does the same. So we wind up with 4 cells. Here, we've got a diploid cell, right? That diploid cell, what's basically going to happen is we're going to put each one of these sister chromatids in their own individual cell. And that does it. 1 cell division is to separate these chromosomes. 1 set of chromosomes go here. 1 set goes in another cell, and then the copies each go into different cells. So we still have haploid cells. The only time there's a diploid cell is when it starts an interphase, where they're copied. Same general things. I'm not going to ask you a lot of questions. I have to remember about what phases occur. Here are some things that I want you to remember. Meiosis produces 4 haploid daughter cells. They are not identical, as was the case in mitosis when the chromosomes were copied. The reason is, in meiosis, you get crossover that starts to occur. In prophase, these start to pair up with their other chromosome homolog. Whenever they separate, they are a conglomeration. In other words, they exchange parts, as you can see, from 1 chromosome to the other. Crossover, these segments, notice how these segments pair up. This one goes there, that one goes there. So we wind up with these exchanges that have taken place. They exchange the same genes, it's just the traits are mixed.
Do we need to remember exactly where they start crossovers? Okay.
Just remember, it actually does occur in just 1 phase. It starts at prophase, but it really doesn't happen until you get between metaphase and anaphase in there. That is crossover. Crossover is responsible for genetic variation. That is why you do not look 50% like your parents and 50%, like each mother or father. You are a blend of their traits. You might resemble one more than the other because of this blend, but you are not 50-50. And what set of genes that you get is just by chance. It's not guided. So don't worry too much about this except for what crossover accomplishes. Don't worry about these phases. The rest of the phases are similar to the phases of mitosis, what happens. And this shows you have forward cells at the end. Still goes through telophase and cytokinesis. They are still pulled to the different poles. But just remember, this is, instead of having a diploid cell where you've got two pairs of chromosomes, each pair is replicated. But then the replicated pair, instead of staying together into the cell, each pair goes into a different cell. So you still have the haploid number now. You go from diploid to haploid. Don't worry about these phases. Remember meiosis 1 and meiosis 2, what happens. In meiosis 1 here, homologs pair up, separate, resulting in two haploid cells. Now these haploid chromosomes have been copied. In meiosis 2, the sister chromatids separate and go into different cells.
Is that all we need to know from meiosis 1 and 2?
Pretty
Meiosis?
much.
Okay.
Now, what are some of the differences, once again, between meiosis and mitosis? Mitosis produces two cells, somatic, with a 2N number, diploid. Meiosis goes through two cell divisions instead of one, as in mitosis, produces four cells that are haploid in number and have been there's been recombination occur with crossover, so the genes have been mixed. They are not exact copies at the end. These are chromosomes that have got variation due to crossover. This is a very good summary right here to study on the differences between mitosis and meiosis. Remember, genetic variation is due primarily to two things. Number one, crossover, and number two, they are random fertilization. You do not know, nothing guides which one of those will be fertilized. Here are the three things. Independent assortment, meaning there is nothing to decide which chromosome goes to what egg. We have crossover occur, and then it is completely random on which egg will or sperm will be fertilized. So remember those three things. There will be a limited amount that I ask you from this. It's mainly the things that I've told you. You're going to see this again when you get into genetics. Chapter 14. Be able to do the Punnett squares. I'm just going to start telling you this instead of going through that chapter. I know what I'm going to ask. I don't need the chapter when I make the exam out. So I'm giving you what I'm going to ask basically. You should know the difference. What makes a dominant trait? What's a recessive trait? You should know what homozygous means, what heterozygous means. You should know that the only way that a recessive trait shows up in complete dominance is if both alleles carry that recessive trait. If it's a homozygous and it's complete dominant or heterozygous, the dominant trait will show up if it's there. If the dominant trait exists in that allele, it will show up. Remember, alleles are the different versions of the characteristic, such as, do you have the characteristic is hair color? You can have an allele that's for black hair, an allele for blonde, brown, whatever. You must be able to do the simple Punnett squares. You must know what true breeding plants are for a characteristic. You must be able to do a simple little Punnett square. Now remember, you can do a simple Punnett square with any combination of alleles. It doesn't have to be pure breeding parents. We can take, for example, this one, this heterozygote right there, and cross it with that homozygous recessive. What you should remember is the result of two pure breeding pairs, all of the F1 generation, the first one, will be heterozygotes. You should remember, if you cross two of those heterozygotes from those pure breeding parents, the ratio that you will get will always be 3 to 1. 3 dominant that shows up, 1 recessive that shows up. So read up on what homozygous and what it is. Remember, phenotype is the way something looks. It's what is expressed. Genotype is what actual alleles that an organism has. Notice the phenotype is purple. Genotype can be homozygous, heterozygous, or heterozygous dominant, or recessive. When we only take one characteristic and do a Punnett square, that is called a monohybrid cross. We can take 2 characteristics, that is a dihybrid cross. You will have to do the monohybrid 4 square cross. You will also have to do a dihybrids. This dihybrids is a pure test cross. It used pure parents. The F1 generation is always heterozygote. If you cross 2 of those heterozygotes, you will always get the 933 to 1. But remember, I may not give you 2 heterozygotes. I can give you any combination. But the way you figure out what goes where, remember, normally females go up and down on the side. Males are usually at the top. Your results will be the same. This is just the traditional way of doing it. One combination that we can have up here is the large Y and the large R that would go there. Then we can have the large Y and the small r that would go there. We can have the small y and the r go there, and we can have the small y and small r go there. So that's the way you set those up. That is the key. If you can set those up, if you look at that and see how to set those up, you've got it. What they're doing is taking these first 2 genes, and what combination can you have with this other gene? So we could have a large Y and a small, or a large R there. We can have a large Y and a small r there. Then we could have this, and that, and that. And that's the way you do the squares. Simple. Remember law of independent assortment, what it is. Each of these alleles segregates independently of that other characteristic. They are not tied together. Note the difference between complete, incomplete, and codominance. Incomplete, the phenotype is going to be a blend. In other words, we have a red flower, we have a white flower. In incomplete dominance, they will be pink offspring for the heterozygotes. If it's homo or complete dominance, it would be red. If in codominance, you might see one petal that was red, one petal that was white. That's where both of the traits show up. This is an example of incomplete dominance, that heterozygote is pink. In incomplete dominance, you've got one dominant, you've got one recessive trait shows up, and then you have the blend. You should understand how blood typing works. I represents the blood type antigen. The superscript at the top represents the version. Is it A or is it B, or I means none. After stasis, a gene at one location alters the phenotypic expression of another. Pedigree is just a family tree.
By the I, the lowercase I on the blood typing, would that just represent O blood?
This does. It represents the absence of either A or B antigen, and that is what O blood would have.
Okay,
Yeah.
You said that I would be the blood type antigen? That I, well, it's representing with the A or
It's
B.
representing that gene.
Okay, thank you.
The A or B is the type antigen.
Okay, thank you.
You should read up a little bit on dominant inherited orders. You should know that in a dominant inherited disorder, many times if it's a bad fatal disease, the individual never reaches reproductive age, so it's not passed on. That's why we have more recessive diseases that are fatal. There are some dominant disorders like Huntington disease that doesn't come on until later age, and there are some that is not fatal, like achondroplasia, the dwarfism. Know about transcription and translation. The basic thing, we're running out of time here, but if you go to know what the triplet codes are and how many amino acids there are, know that a codon is 3 nucleotide sequence. This shows how messenger RNA is made. Should have an idea of that. The initiation, elongation, and termination. Should know what the Tata box is. And at the end of this, remember it's RNA polymerase that makes messenger RNA. Remember we have start and stop codons. We have a poly A tail, polyadenylation signal. Introns are the code, or the part that don't code. Exons are the part that do. So the introns have to be cut out and then the DNA spliced to get a functioning messenger RNA. Before that happens, it's called pre-messenger RNA. That splicing occurs in the spliceosomes. Ribosomes. RNA is an example of RNA can act as an enzyme. They are called ribosomes. They do the splicing of messenger RNA. This is the sample of the ribosome, and now we've got tRNA that comes in. EPA, remember, is these are the sites and what they do. And if we look at this, this gives you a good little version. Comes in on the A site, delivers its amino acid at the P site, exits then at the E site. And we have the glory peptide chain. This is a good summary of transcription and translation right there. If you understand what's happening right here, then you understand transcription and translation. It's the best thing in all of it, really, to study. And what we went over and finished up with on DNA replication. Test will be pretty much the same. Now let's bring any of your study sheets, groups that you had, one per group, everybody listed, and how much each individual person was there. Turn those in with the exam on Friday at 1:30 in this room, okay?